Hey you guys, welcome to chapter 12.1 and 12.2. We are talking about the cell cycle. And so we start with the cell theory, or part of the cell theory, which states that every cell comes from a cell, comes from a pre-existing cell. So there is no spontaneous generation. A cell doesn't just come from nowhere. And this is important because back in the 1800s, when scientists like Rudolf Virchow were studying biology, they thought that, or people thought that, rats came from the tablecloths in the corner of a room because people saw rats running out from behind the tablecloths. So that made sense to them, that rats came from tablecloths. But we now know that rats come from rats and cells come from cells. And cells and life comes from life. And so life has to come from life. And it does this through cell division. And if it's a single-celled organism, cell division is a form of asexual reproduction. However, if it's a multicellular organism, it's used for growth or for regeneration. And so the primary function, or one of the primary functions of cell division, is dividing up the genetic material. So we have to, if within a cell, we have to split up the DNA. So when the cell splits, half the DNA goes into one cell and the other half goes into the other cell. And so the entire genetic complement within a cell is called the genome. And all of the cells in your body have the same genome. So the nucleus enclosed genetic material within your cells is going to contain the same genome. The same genome that's in your toes is the same genome that's in your nose. And so, like everything else in biology, there is about 5 words that mean very similar things. And so chromatin is the complex of DNA and proteins that helps it to coil up. And so we can see a picture of that here. However, chromosomes is a similar word, or has a similar meaning, but it's just more specific. So when we're talking about your first chromosome or your second chromosome, it's a specified length of chromatin. So your chromosomes are made up of chromatin. And they contain hundreds to thousands of genes. So when we're talking about one specific strand of chromatin, or one chromosome, at least for us, human beings. And for each eukaryotic organism, there's a set number of chromosomes. So we humans have 46 chromosomes in our cells. We have 23 from our moms and 23 from our dads. So this is a karyotype, and we can see the 46 chromosomes. We know that this is a female because there's 2 X chromosomes. And so this is your first chromosome, and this is your second first chromosome. This is your second chromosome, and this is your second chromosome. And we have 46 of these, or I'm sorry, 23 pairs. And somatic cells contain these 2 sets, so one from mom and one from dad, 23 from mom, 23 from dad. However, gametes, these are sperm and eggs. These contain half the genetic material because the sperm and the egg have to come together. And so we need to have HALVE, the amount of DNA in there, so that when fertilization occurs, we're not doubling up every time. So our gametes have half the amount of DNA, or half the number of chromosomes. When we're talking about chromosomes, we need to think about the cell cycle, so the life cycle of a cell, and what the DNA or what the chromosomes are doing at each phase. And so the phases of the cell cycle, one big long phase that contains other phases is interphase, and that's 90% of the cell's life, or the cell cycle, is a cell is in interphase. Interphase is made up of G1, S, and G2. So G1 is the cell is growing because here it's a brand new cell, and so it is smaller, and so it's going to grow larger. It's going to make more organelles. It's going to make more proteins. It's going to come into itself, and it's going to begin to go into S phase, which is when the DNA is copied. So G1 is growth. It's getting ready for synthesis, which is DNA replication. And here the DNA is replicated. And then G2 is when we are getting ready for the M phase. So once again, growth, because the cell needs to get larger because it's going to split in half, making more organelles, making more proteins because the cell is going to divvy everything up in half. And then we go into M phase, which is the cell division or division of the genetic material and cytokinesis. During interphase, the G1, S, and G2, our DNA is more uncoiled. Our chromosomes are going to be varying levels of coiled throughout the cell's life, throughout the cell cycle. Here, they're going to be more uncoiled because we need to access the genes. We have enzymes that need to access the genes so that we can transcribe them and we can make proteins, because we're making proteins during G1 and G2, a lot of them. So during interphase, the chromosomes tend to be coiled up and then, uncoiled, and then during the M phase, it coils up and it gets very organized so that it can be divvied in half in a very specific way. So during G1, we're going to have we have 46 chromosomes in a human cell, and there's going to be just one copy of each of the 46 chromosomes. Then we go into synthesis, and we make the second copy. We make an exact copy of it. We call these sister chromatids. They're exact copies, and so we have 46 chromosomes, each with an exact copy. So if you think about it, it's like having 93 chromatids, 93 individual little chromosomes, but each chromosome has an identical sister chromatid, and so there's 46 of those. And each of the sister chromatids are attached to one another with proteins called cohesions. And then right here in the middle, this is the centromere, and this is a section of repeating sequences, which is sort of the waist right here of the chromosome of the two sister chromatids, and it's just more closely bundled. This is the place where it's most closely attached. So this is the waist, and these coming out, these are the arms, and so these are our chromosomes, or our sister chromatids. Before we start talking about the actual cell division, we need to talk about spindle fibers because they play a large role in the process of mitosis, or the process of cell division. We know that interphase is about 90% of the cell life, or the cell process, and so therefore the M phase, or the cell division phase, is about 10% of the cell cycle, and it consists of mitosis and cytokinesis. The difference between mitosis and cytokinesis is that mitosis is the division of genetic material only, whereas cytokinesis is the division of the cell. So first we divide the genetic material, and then when we have two separate nuclei, or two separate sets of genetic material, then the cell can split, or then the cell can go through cytokinesis. And these events of mitosis, or division of genetic material, are heavily dependent on the mitotic spindle, which is made of microtubules and proteins. So when the mitotic spindle begins to polymerize, other microtubules in the cell begin to break down, so the pieces are sort of taken from other places, and it's going to elongate. So we have the mitotic spindle is going to grow. The microtubules are going to have addition of subunits onto the growing end, or onto the positive end. When it shortens, these subunits are being removed. There's a good little video right here to help you visualize this process. And I recommend checking this out before we go into the rest of this, because it will really help you to sort of understand everything a little bit more clearly. The assembly of the microtubules, or the spindle fiber, is going to start at the centrosome. And this is a word that people get confused with because it sounds so much like centromere. And so I like to remember this because there are some centrosomes. There's a couple of them. There's some of them. So centrosome of them, a couple of them. Whereas the centromere, there's merely one at the middle of the chromosome. So there's some of them. And so the centrosomes generally hang out by the nucleus, And then as the cell goes into M phase, they're going to move towards the opposite ends of the pole, and then microtubules are going to start growing out of them. And as microtubules start growing out of the centrosomes, they take on this sort of star shape, and we call those asters. And they extend from the small microtubules that extend from the centrosomes are called asters. And they grow into spindle fibers as the subunits attach, and they attach to the sister chromatids at the kinetochore. The kinetochore is in the centromere region, that center region of the sister chromatids, and it's a region of proteins. And so the microtubules are going to attach to these region of proteins, and we call this the kinetochore. This is going to sort of play a tug of war. These spindle fibers are going to play kind of a tug of war with the chromosomes, and it's going to move them to the metaphase plate. So if we're looking at the cell, the chromosomes are going to be lined up at this imaginary plane called the metaphase plate. And at that point, an enzyme called separase cleaves the cohesions, and then the sister chromatids can break apart. They can start moving to the opposite poles. They can migrate to the poles. And the mechanism of the spindle fibers moving towards the poles is not well understood, but it is thought to be a function of both motor proteins walking back towards the centrosomes, and then motor proteins reeling in the chromosomes or depolymerizing. So if this is the microtubule, and here's our motor protein, and it's pulling the microtubule back, and you can see that the subunits are starting to come off, and so it's depolymerizing. We think of this process as the chromosomes moving towards the poles as like the microtubules is sort of pulling these chromosomes apart, but it's not necessarily like that, and it's different in each species. But the motor proteins are sometimes walking them back, kind of moving on their own, and sometimes they're being reeled back. It's a topic that is still being studied today, so there's lots to learn. We are at the end of interphase, G2 phase. Remember that we're getting ready for the M phase here. We're getting ready to divide the cell, to first divide the genetic material and then divide the cell itself. And so during G2 phase, the nuclear envelope encloses the nucleus, so we're just regular cell life. The genetic material is uncoiled, so if you look at it under a microscope, it just looks like one blob. You can't see independent chromosomes at that point. It's just, they're too small, so it just looks like a blob. And the chromosomes are duplicated, or I'm sorry, the centrosomes are duplicated right here. So these are replicated, and they're hanging out near the nucleus. And we're getting ready to go into M phase. So during M phase, our first step of M phase is prophase. During prophase, chromatin fibers begin to coil. If we look at this under a microscope, we can start to see independent chromosomes because they start to get thicker. The nucleoli break down and disappear. In the mitotic spindle, these microtubules begin to form, or asters start to spring out, and the centrosomes move away from each other. So they're moving away from the nucleus, and they're starting to move towards opposite ends of the poles of the cell. During prometaphase, our nuclear envelope starts to break down, and the microtubules are extending from the centrosome, and they start to break into that area where the nucleus was, and they're starting to attach to the chromosomes, or the sister chromatids, at that kinetochore right here. And there are, if you can see right here, non-kinetochore microtubules. They grow and they start to attach to one another, and they push against each other. So they're not attached to any chromosomes, but they're attaching to one another, and they sort of push the cell apart so that the cell begins to lengthen, which is going to allow for the cell to split in a little bit. During metaphase, I like to think of metaphase as the middle. The chromosomes are in the middle during meta. They both start with M. So centrosomes are at the opposite ends, and the chromosomes are in on that imaginary plane, or in the metaphase plate. And we are getting ready for anaphase. I like to think of anaphase as a part. They both start with A. During anaphase, the chromosomes separate and they move away from each other. What allows this to happen is the separase is going to cleave the cohesion, so the sister chromatids can break apart and start moving away from one another. The chromosomes move towards the poles while the microtubules shorten, and the non-kinetochore microtubules lengthen and elongate that cell. By the end of anaphase, we have two genetic complements. So the genome, the entire genome, is on one side, and then a second copy of the genome is on the other side of the cell. So we have two copies of the genetic material within one cell. Telophase is our last phase, and here we haven't split. We still have not gone through cytokinesis. We have two nuclei forming. We have our nucleolysis forming. It's just the opposite of prophase. The nucleolyses are reforming, and the chromosomes start to uncoil because they're getting ready to go into that G1 phase where we need to make lots of proteins, and so the DNA needs to uncoil so that it can be accessed and read by enzymes. So the division of genetic material at this point is complete. We have one cell with two nuclei, and then we go through cytokinesis. Cytokinesis is the division of the cytoplasm. That's all of the stuff that's in the cell that's not the nucleus. We split in half the organelles, the proteins, etc., etc. So in animal cells, a cleavage furrow appears. We can see that right here. And that, what happens is the microfilaments, that's the thinner type of cytoskeleton, and it wraps around and it just sort of pinches off. I like to think of a balloon, and if you were to tie a string around the balloon and pull it, it would sort of pinch that balloon into two, and that is essentially what's happening in animal cells. In plant cells, however, a cell plate is formed. So what happens is vesicles from the Golgi apparatus move over to the side where it just separated, and it builds sort of a scaffold for which the cellulose is going to be able to attach to. So that is the cell plate. It's the sort of scaffold for the cell wall to build on. And that's the end. Let me go back really quickly. That's the end right here of cell division. So at that point, we have two brand new cells that are moving into G1 phase and starting all over. And that is how eukaryotic cells divide. What we're going to look at, just one slide, is binary fission. This is how prokaryotic cells divide, or single-celled organisms. This is asexual reproduction of single-celled organisms. And essentially what happens is the chromosome, there's one single loop of chromosome, and it's going to divide. So here's the origin of replication. And it's going to start to replicate, and one of the origins is going to go over to this side, and the cell is going to elongate and elongate, and the chromosome is going to continuously be replicated, and then when the replication is finished, the cell is going to divide. It's just a very much more basic form of cell division because there's no nucleus and there's fewer chromosomes, and so it's just a more simple process for a more simple type of cell. And those are the notes for 12.1 and 12.2.